Hey, Dr. Segan here, and today we're going to be doing another virtual lab. This one is going to be about microbial diversity. And to study microbes in a lab setting, you have to use our friend here, the microscope. So before we get to looking at some microbes, we need to learn a little bit about microscopes and how we can use them as tools to identify microorganisms. Well, the first thing I think that everybody knows about microscopes is that they allow you to see things that you cannot see with your normal eyes. They do this by magnifying images. And the way this magnification works is you have an eyepiece here that is set to a certain magnification, and then you also have these things called objectives here, and you can rotate this part to basically zoom in and out using this microscope. When we're talking about the total magnification of a microscope, for looking at this is the eyepiece. This actually magnifies things by a factor of 10. So just looking at something through this eyepiece will magnify it so it's 10 times larger than it was previously, and usually that's marked with something like a 10x symbol. And each of these objectives also has a number on it, which represents their kind of factor of magnification. This red one, I don't know if you can read that. It says 4 right here, 4 over 0. Let's see if I can get that bigger. This red objective has a magnification factor of 4. The eyepiece has a magnification factor of 10. If you take 4 times 10, that's 40. So when I look at something through this red objective, it appears 40 times larger than it would in normal life. This yellow objective here, I don't know if you can see that, it says 10 next to it, so this is a 10 times magnifier. If you go 10 times 10, when looking at objects through this one, things are 100 times larger than they would be in normal life. And this next one, which is usually the highest I ever really can go with this, it says 40. So if you go 40 times 10, that's 400 times larger than something would be in real life. And you can see really, really tiny things like bacteria with that. There is a 100 times magnifier here, objective, which would mean 100 times 10 would make it a times 1000 magnification. But these are generally pretty tricky to use, and usually you need to do something called oil immersion with them, and I usually don't mess with that. So for the most part, we only ever go up to 400 times magnification for our microbes. So what can you actually see at 400 times magnification? Let's start with looking at a few bacteria here. These are dead bacteria that are stained on a slide, and we'll see what these bacteria look like through our 3 objectives that we're going to use. So we'll look at bacteria with the 40x, the red one, the 100x, the yellow one, and then the 400x, the blue one. Let's see what it looks like. All right, so this is at 40x, and it was a little blurry, you couldn't see much anyway, and now this is at 100x. See, these little red stringy things are actually dead bacteria. Those are what bacterial cells look like. Not much to look at. At 100x, we can get the best view of them. And pardon a little bit of gray stuff on there, but this is that's bacteria, these little stringy things. And these are actually clusters of cells. The tiniest little red dots you can see are actually in bacterial cells. So from that, we can see that there's a huge difference between 40x magnification and 400x magnification. And we can also get a perspective that these things are unbelievably tiny. I mean, this is the slide. Looking at them at normal magnification, 1 magnification of your normal eye, there's absolutely no hope of seeing those tiny little creatures. So that brings us to our next topic of, okay, if we actually need to identify different microbes, one of the most useful tools to do that is to know exactly how large or small it is. there is a huge difference between the size of a typical protist and a typical bacteria, for example. And we can use our knowledge of magnification in these microscopes to figure out how large different things are. But before we do all that, just so you can have an idea of this range of sizes of microbes, how big and small all these things are, all in the microbial world, and how much diversity there is down there, let's watch a short video about the sizes of these organisms. Right, so let's take a brief moment here and explain the units that are being used in this video. The unit here is a micrometer, and that's this weird little symbol that you see, kind of U-imish looking symbol. Micrometers, or also known as microns, represent a unit of metric measurement in the metric system. A meter is, I think, a meter is this long. This is a meter stick. This is 100 centimeters, since you're referring to 100. And I think we're all pretty familiar with the idea of a meter. If we divide a meter by 1,000, that gets us a millimeter. And a millimeter looks like those little marks right there. So these are millimeter marks. A micrometer is if you took one of these little marks and divided it by 1,000. So you can imagine if you took this little mark right here and divided it by 1,000, you would have 1,000 micrometers or microns. When we're talking about our smallest viruses, like COVID or the flu, we're talking smaller than a micron. So we're talking about, basically you could fit about 10,000 COVID or flu viruses in just one of these tiny little spots here. So you could fit, if you lined them up end to end, you could fit 10,000 COVID viral particles in a row in that little space there. So unbelievably small. And that gives you an idea of perspective for everything else that you're going to see for the rest of this video. All right, so hopefully that gives you some perspective on the different sizes of these organisms. And it also kind of begs the question of, okay, how small of creatures can we see on this microscope? We just saw some bacteria, and that means that we can see things on the scale of microns, but not very clearly with this microscope. And unfortunately, this microscope is not powerful enough to actually see something like a typical virus. It would just be too small to even see with this microscope. But you can also see the importance of knowing how large the creature you're looking at is in terms of identifying it as a microbe. There's a huge difference between the size of the average protist and the size of the average bacteria. So with that in mind, the next part of this lab is going to involve figuring out how to make reasonably accurate measurements of the things that we're seeing under this microscope. And to do that, it's actually really simple. I'm sure there's fancier mathematical ways to do it, but the simplest way to kind of visualize it is I have made on this slide a couple of tiny little marks. These marks are a millimeter wide, and they're spaced a millimeter apart, and they're exactly 3 millimeters long. And so we can put this under a microscope and see what it looks like, and that'll give us an idea of the sort of scales that we're seeing on the microscope. For example, if this takes up the entire view screen, we know that the entire view screen is about 3 millimeters long if that whole line takes up the entire view screen. So let's see what this looks like under these different magnifications. All right, so you can see that when we zoom in and look at these little lines here, at the 40x, you could pretty much, the field of view would be right around the size of these lines, a little bit bigger. And the field of view for the 40x is about 3.5 millimeters. So you can really get in there and see what's going on at that scale at 40x. If you're looking at a microbe at 40x, more than likely, if you were looking really carefully, kind of like we are here, you could probably see it with your naked eye. Things that are visible at 40x, yes, you can see them much more clearly, but technically those aren't even microscopic. Once we zoom in to 100x, we could see that the width of one of these lines pretty much took up the majority of the field of view. And at about 100x, the field of view is, I think, around 1.5 millimeters. And when we zoom in all the way up to 400x, you can't really see much of anything. It's just kind of all black, and each of these lines actually takes up the width of one of these lines takes up the entire view. And that's less than a millimeter. The entire view screen is less than a millimeter. So that means 1 millimeter is 1,000 microns, so we're less than 1,000 microns. And actually, the full view would be something like 750 microns. That's my estimate for the fully zoomed in magnification. We can use these numbers that when we take recordings of microorganisms, and if you want to measure them pretty accurately, generally you have to take recordings of them, some kind of picture or digital video or whatever. We can superimpose those field of view measurements over the recording to get an idea of how large the microbe is. For example, if we wanted to measure how long this mouse was, we could just line it up to a ruler. We can kind of do the same thing with a digital recording where we basically put a ruler over the top of whatever microbe we're trying to measure and figure out just exactly how big it is. And knowing how big it is will help us identify the microorganism that we find. So let's go have a look at some different microorganisms, talk about their characteristics, and also measure how big they are. Let's see what we can find. Where you have some pre-made slides, we have a sample from the grocery store, and we also have some bayou water, basically water from the lake down the street, the Willow water hole. We'll see what kinds of things are swimming around in there and see if we can identify them. All right, let's get to it.